Oh my goodness.
Looks good.
Kentucky, finally.
All right. It's like, well, let them know that PowerPoint. Well, folks, I think we're recording some sounds here. Yeah, it looks like we are. There obviously were a few issues in the first couple minutes this morning, but we're ready to go, and it won't be long before somebody from CATS comes running in here to try and fix it so that the sound works, even though it seems as if it is working right now. So, how you guys doing? We're in the home stretch, right? I'm counting down the number of lectures. I'm sure you guys are as well. I've gotten a couple of questions now about the impending final exam for the class. Let me say a few things to you about it again. I've said this to you already, I believe, but we'll do it one more time, and I'll probably do it another time or two next week. So there is a final exam for the class. No big surprise there. The registrar determines what time the final exam will be, and the registrar determines the time like years, literally years, before the class begins. And for this class, the final exam, it will be on Tuesday, December 13th. I'm pretty sure, correct me if you think I'm wrong, but yeah, so Tuesday, December 13th from 8 a.m. until 10 a.m. It'll be right here in this room. You won't need 2 hours to take that final exam. I think that 45 minutes would probably suffice for most people. You know, an hour would be luxurious. You can go and double check your answers a couple times if you like. There's nothing else going on in here that day. So when you're done, leave, right? No reason to stick around. And we'll have the grades posted on Pilot for the exam probably later on the 13th, and final grades will probably be taken care of shortly after the final exam scores are posted. The exam is going to be multiple choice, just like the 2 midterm exams that you've seen so far, and there will be 58 questions. 50 of those questions are going to be from stuff that I've talked with you about just since the second exam in the class. So for the last couple of weeks, and today and next week. So 50 of the 58 come from stuff that I've only talked with you about since the second exam for the class. The other 8 questions are cumulative. And so what I'm going to try to do is ask you an equal number of questions from each of what I see as the 3 parts to the class, right? So the stuff leading up to the first test, stuff leading up to the second test, and the stuff leading up to the final. And if you stop and you think about it, what that means is that 52 or 53 of the 58 questions are going to be about things that I've talked with you about just since the second exam. And then there will be 2 or 3 questions from both the first part of the class and the second part of the class. Okay? So 58 multiple choice questions. Bring a number 2 pencil. It's all going to be Scantrons like we've used so far this semester. There is a review session that we'll have off the top of my head. I don't recall the particulars of it, but it is spelled out in the syllabus. I know it will be on the day before the final exam, so the Monday before the final exam. I think it's from 10 till 12 o'clock in the morning on the Monday before final exams. That is a a dead spot on the final exam schedule. No final exams are scheduled for that particular block, so there shouldn't be conflicts. If you'd like to come to this optional review session, you're more than welcome to. My recollection is that it is not in this room, but the room that it's in is spelled out in the syllabus. I'll mention it to you again next week sometime. It's an optional review session. I will seriously answer any question that you put to me. I don't care. It can be about what my shirt size is. It can be what my favorite beer is. It could be about how the lac operon works. I'd enjoy talking with you about any of those topics, some a little more than others. But any question that you have, I'll be happy to answer. I don't mind answering the same question 2 or 3 times. So if you come late and you ask a question that somebody else has already asked, I am not going to berate you for coming late. I'll just answer your question, and you'll have to figure out why everybody around you is snickering, because I'm probably saying the same thing that I'd said before you had come in. So optional review session, and I want to emphasize the optional part. It's for you guys. It's sort of like just an open house office hour in my mind. If you want to come, you're welcome to. Don't feel like you have to, or that there will be any secrets that are revealed. I'm just going to answer questions that people ask me. All right? So final exam coming up. Not too soon to start looking at those old exams on the pilot page of the course. In fact, it may actually be starting to be a little bit late to start looking at those final exams on the pilot page for the course. That'll be your single best way to prepare for that final exam on Tuesday, December 13th. Okay, the administrative stuff is taken care of. Let's get to the fun part, talking about the regulation of gene expression. You guys got to think I'm nuts, you know, because I really have fun talking about things like the regulation of the lac operon. Let me tell you that the stuff that I told you about in lecture on Tuesday is stuff that I think is about the coolest stuff that I can share with you. It's actually about 90% of what makes the lac operon cool. The next 10%, the rest of the story, if you want to stop by and talk with me after class or in my office hours or something like that, I'll be happy to spill my guts to you and tell you the rest of the story. But it's beyond the scope of this class, and it won't come up really until you take a sophomore level course in the fall probably of next year, Bio 2110. But there's some other neat little gizmos that are playing out in the lac operon. I'll be happy to talk with you about it, but again, what I told you about on Tuesday, I think by itself is a heck of a story. It's tough to beat that story. So there's a picture that summarizes the lay of the land for the lactose operon. I wanted to start today just by pointing out a couple of details to you that may have been lost as in my excitement to share with you some of the coolest aspects of the lactose operon. Let's take just a couple minutes to make sure you're seeing some of the details associated with a picture like this. And wow, that's a really small point. But hey, I tell you what, I think I'm going to be able to write on the slide. So it's a nice trade-off. So here's the lac operon, the lay of the land, right? Remember, there are these 3 structural genes. That's what they're called. They code for proteins that float around and do things that need to be done to metabolize lactose. There's the lac-Z gene, the lac-Y gene, and the lac-A gene. Z codes for the protein that breaks lactose into its 2 component monosaccharides, glucose and, I believe, galactose. Permease is the protein that lets lactose get moved across the phospholipid bilayer of the cell, right? So from outside the cell to inside the cell. And transacetylase puts acetate groups on that lactose once it's inside the cell so that it can't get out the same way that it had come in. Notice that all 3 of these genes are head to tail, one after the other. That's actually a neat design feature. That's an important detail. It's a recurring theme for a lot of prokaryotic genes. Genes with related functions are often clustered very close to each other and work as part of a single operon. Okay? And what's neat about that is that not only is it sort of anal retentive, it's like an extremely ordered way to set up this system. What's especially neat about that is that all three of these genes share a single promoter. And what that means, in fairly simple terms, is that if you express one of those genes, you're going to express all the rest as well, right? So can you think of a circumstance where it would be helpful to express the beta-galactosidase gene and not also express the permease gene in the transacetylase gene? I say that somewhat facetiously because I think the answer is fairly obviously no. If ever you're going to make beta-galactosidase, the gene that breaks up glucose into its that breaks up lactose into its two component sugars, if ever you make that protein, of course you also need the protein that helps bring lactose from outside the cell to inside the cell, and you also need the protein that keeps lactose in the cell. In a word, and there is a word that describes this. This is coordinated expression, right? Good word, right? If you express one gene, you express the others. If you express one gene, you express the others at the same level even. Pretty cool, huh? Nice design feature, clever way to have set this up. And again, this is a recurring theme for a lot of prokaryotic genes. They tend to be genes with related functions tend to be clustered together. They share a single promoter that assures their coordinated expression. And boy, you know, how could you have hoped or designed a better system than that? So, 3 genes. Now, some fancy words for you here. You can impress your friends with these vocabulary words. Cistron is a Greek word that describes a cohesive chunk of information. Okay? The information you need to spell out the amino acids to make the beta-galactosidase protein can be thought of as a cistron. It's a cohesive chunk of information. And so you could call the beta-galactosidase gene itself a cistron. Where it becomes interesting or useful is to talk about the messenger RNA that gets made from that operon as being a polycistronic message. Poly, multiple, cistron, cohesive chunk of information message. That messenger RNA has, in this instance, 3 cistrons, 3 chunks of information. Prokaryotes, because of the way they organize their genes in these operons, typically have polycistronic messenger RNAs. Eukaryotic genes, and that's where we're going later today, eukaryotic genes are exclusively, they are invariably monocistronic. We only make one cohesive chunk of information from our transcripts, but prokaryotes have got this worked out, and they're working with polycistronic messenger RNAs. But here are some of the details that I imagine you didn't notice during the lecture on Tuesday. Let's focus on them for a little bit here. See that little green box here? That'll make it easier to see. See that little green box on that messenger RNA? Right? That's what that pinkish thing trailing behind the RNA polymerase here is supposed to be. That's a messenger RNA. That green box is supposed to make you think, ooh, that is a start codon. Okay? You know what start codons are. You may actually know what a start codon is. I happen to know. It's AUG. There are 3 nucleotides there, AUG. That's what a ribosome is going to look for to begin translation of the downstream gene. Okay, what's that downstream gene? It's the beta-galactosidase gene. So the first amino acid that gets brought into the new beta-galactosidase protein that's going to be translated by a ribosome is a methionine. The codon that codes for that methionine is right there. That green box is a start codon. Notice that there's some pink stuff happening before that green box. The 5' end of this messenger RNA is right there. You may also happen to recall that that's going to be an A. The first nucleotide, the transcriptional start site, the +1 nucleotide, all equivalent terms, is always an A. There's an A here at the 5' end, and then there's other stuff before you get to the translational start site. I gave you guys a word for that about 2 weeks ago. That's what we call the 5' untranslated region. It's transcribed, but it's not translated. Okay? I told you it's sometimes called, the 5' UTR, the 5' untranslated region. U for un, T for translated, R for region. So there you go. Lay of the land here is you got a 5' UTR there. You got a start codon. What comes after the start codon? Well, in a word, an open reading frame. I don't know if that's the right way to say that in 3 words. It's an open reading frame. In 1 word, hip people would call that an ORF for open reading frame. All right? I'm serious. Hip molecular biologists. You know, I call that an ORF. Yeah, you want to be hip? Go ahead. Call it an ORF. People will know what you mean. That's exactly what this is. It's an open reading frame. And you know what's at the end of the... You guys are laughing. Yeah, what's going on? Yeah, at the end of the open reading frame is a red box. Okay? You know what a green box means now, if you hadn't before. I think you probably don't need me to tell you that a red box is a stop codon, but I said it anyway. All right? That's what's going to come at the end of the open reading frame. So everything between the green box and the red box are triplet codes that spell out specific amino acids that need to get strung together by ribosomes during translation to make the specific protein that does what beta-galactosidase does. Pretty simple. What's after the red box is a green box. You know what green boxes are, right? Green boxes are start codons. Check this out. The red box is right up against the green box. That's not artistic license. That's the way these things work out with prokaryotic polycistronic messages. You'll have a 5' untranslated region at the beginning, and then you get to a coding region, an open reading frame, and at the end of that open reading frame, not very far away, will be the start codon for the next open reading frame, for the next cistron. We're not wasting space here. We're not wasting nucleotides at the level of the RNA. We're not wasting nucleotides at the level of the DNA. This is very densely packed at the level of the DNA, and that in turn ends up being densely packed information at the level of the RNA. Prokaryotes are all about saving ATPs. They want to save energy. If you've got a whole bunch of wasted space between the end of one open reading frame and the beginning of the next open reading frame at the DNA level, every time you replicate DNA, you're burning ATPs. Every time you transcribe this polycistronic messenger RNA, you're burning ATPs. There's pressure on prokaryotes to be as streamlined and energy efficient as possible, and that shows itself here in the very close proximity of the stop codon for the LAC-Z gene and the start codon for the LAC-Y gene. I'm not kidding when I say prokaryotes are all about saving ATPs to the extent to which they put these right up against each other. Now, it may be a little bit of an exaggeration, or maybe it's a matter of scale. There will probably be 10, 15 nucleotides between the stop codon and the start codon. But the point still stands. They're really close together. You're not wasting space. You're not wasting ATPs. What comes after this little green box here? It's a start codon, AUG. It's the methionine that is the first amino acid for the LAC permease gene. After that is, come on, you be hip with me here, it's an ORF, right? It's an open reading frame. It's a long run of triplet codons without there being a stop codon. The red box there is a stop codon. So there you have it. That's pretty much the lay of the land. You know, big picture, sort of conceptually, what's going on here is this, is you've got a promoter. I told you on Tuesday the promoter is effectively 13 nucleotides. There's the +1 nucleotide, that A. At -6, there's a TATA box. At -35, there's a PRIBNOW box. PRIBNOW and TATA both are about 6 nucleotides. Plus that 1, you get a total of 13. There's an orientation to those nucleotides so that when the polymerase comes along and says, ooh, this is a promoter, that also gets the signal, and the coding sequences are that way. They're downstream. It's not just that there's a gene nearby. There's a gene nearby, and it's over there. Okay? And that's pretty much all that the polymerase is looking for. It's looking for those 30-some nucleotides, 13 of which are being scrutinized upstream of the transcriptional start site. Just downstream of the transcriptional start site is the sequence of about 20 nucleotides that the LAC repressor binds to. Notice that's part of what's going to show up in this 5' untranslated region. It doesn't play any role in the RNA level, but a repressor protein can bind there and prevent RNA polymerase from moving downstream. Okay? When a repressor protein binds to that specific nucleotide sequence, it interferes with the activity of an RNA polymerase. We call that negative regulation. It's negative regulation at the level of the initiation of transcription. There are other types of negative regulation that can take place, but that's pretty much the single best way to regulate a gene is at the very start, at the level of the initiation of transcription. That regulator is called the LAC repressor. It's the LAC-I protein coded for by this gene over here. When it's a two-trick pony, right? When lactose levels are low, it hunkers down and it prevents transcription. When lactose levels are high, it binds to lactose, its other trick, and it falls off the operator and it allows transcription to occur. I dropped on you in the last 5 minutes of lecture on Tuesday the idea that the LAC operon also is like the prototypical classic example of positive regulation of gene expression. It turns out that the promoter sequences for the lactose operon don't do a very good job of attracting the attention of passing RNA polymerases. They're off by 3 from what it is the RNA polymerase is really looking for. That deficiency gets made up by a positive regulator, cyclic AMP receptor protein. Cyclic AMP receptor protein binds to nucleotides in the promoter of the lactose operon, but it binds only, I'm thinking at the very least I should be able to make these dots bigger. Kind of looking for arrows. Well, I'll just have to wave my hand with my little tiny dot today. Can you guys even see my little tiny dot? Maybe I need to highlight stuff. Cyclic AMP receptor protein only binds when glucose levels in the cell are low. Okay? And when cyclic AMP receptor protein does bind, RNA polymerase says, hey, I like hanging out with cyclic AMP receptor protein, and you know, I'm being kind of colloquial or just chatty here. You guys know what I mean, right? There's stuff on the surface of cyclic AMP receptor protein. There are amino acids poking out, right? There are amino acids on the surface of the RNA polymerase that like rubbing up against those amino acids that that are found on the cyclic EMP receptor protein. It's one of those kind of things that's going on here. Hydrophobic, hanging out with hydrophobic, positive, hanging out with negative. Cyclic EMP receptor protein has on its surface amino acids that RNA polymerase really thinks are cool, and it wants to check out and spend more time with. When cyclic EMP receptor protein is there on that promoter sequence, RNA polymerase will come around to check it out, whereas it might not have been that interested in sticking there just on the basis of the nucleotide sequences themselves. But if cyclic AMP receptor protein is there, that means that glucose levels in the cell are low, and it's in those circumstances that you want to consider expressing the genes of the lactose operon. Glucose is the preferred sugar for prokaryotic cells in general, E. coli in particular. If glucose levels are high, don't bother with the secondary, less preferred sugars. Just use all the glucose and be happy. If glucose levels are low, now's the time to start thinking, is there some alternative to glucose that's around? If glucose levels are low and lactose levels are high, those are the circumstances when you might want to express the lactose operon. All right. Now, on Tuesday, I told you that I wanted to tell you today about another operon, just to sort of give you a rounded perspective here. I think I mentioned the tryptophan operon. Let me introduce you to the tryptophan operon. What I hope you'll hear as I talk with you about the tryptophan operon is look for the things that are similar and look for the things that are different from the lactose operon. There's an awful lot that's similar. And I hope that that conveys to you that what you know now about the lactose operon pretty much tells you the story of the couple hundred other operons that E. coli works with. All right? So figuring out, knowing what's going on with the lac operon by itself tells you the story for pretty much all the rest. So, the lac operon. It is all about metabolizing lactose. That's it. It's not good for anything else. It metabolizes lactose. You can be a little more specific, I suppose, and you can say that the lactose operon is a catabolic operon. It breaks down lactose. Here's a difference. The tryptophan operon is an anabolic operon. Okay? Well, the lac operon is all about breaking up lactose. The tryptophan operon is all about making tryptophan, building it up. It's an anabolic process as opposed to a catabolic process. But bacteria, like E. coli, sometimes find themselves in the circumstance where they would like to have more of the amino acid tryptophan than they currently have. You can imagine why. Tryptophan is one of the 20 amino acids. It plays important roles in the structure of some proteins. There's sometimes that only a tryptophan will do if you want to make a protein that has a particular function. If tryptophan levels are low, it would be a good idea for a bacteria to make some of that tryptophan. Right? If it can get it from its environment, swell. That's free. It'll be happy to suck it up from outside. But if there is none outside and it's used up all of its tryptophan, the time has come for it to buckle down and make some tryptophan. And how do you make tryptophan? Making tryptophan involves the expression. To be able to make tryptophan, E. coli has to be able to produce 5 proteins. Guess where those proteins hang out at the level of the genome of E. coli? They're part of a single operon, right? No big surprise there. Same thing we were talking about for the lac operon. We've got 5 genes with a related function. They're involved in the stepwise synthesis of tryptophan from less complicated molecules. Those genes are named TRPE, D, C, B, and A. They code for proteins that when they're working the way that they should, let you make tryptophan out of simpler starting molecules. Fair enough? check it out. They all share a single promoter. They're part of an operator, right? So they have one promoter. Boy, I'm not good with my circles, am I? They've got one promoter. That's going to assure coordinated expression. If ever you make the TRPE protein, you're also going to make the TRPD protein. Makes sense. No point in starting to make the tryptophan molecule and not finishing the job. Right? They share a single promoter. You're going to have coordinated expression. They've got an operator sequence, just like what you were seeing with the lac operon. Just downstream of the promoter is a set of nucleotides to which a repressor protein can bind. There's a repressor protein. Here it's called the TRPR protein. Okay, lac operon, it's lac-I. For inhibitor, here it's TRPR for repressor, the TRP repressor protein. When do you want to have the protein product of the TRPR gene bind to the operator of the tryptophan operon? What makes sense? It's when it binds, I'll tell you, it's going to be a negative regulator. It's going to stop the RNA polymerase from transcribing the downstream genes. Right? When do you not want to express the genes involved with tryptophan biosynthesis? If you think this is a hard or a trick question, I have bamboozled you somehow, unintentionally. It shouldn't be that hard. It comes down to this. If you got a lot of tryptophan, don't make any more. Right? Fair enough? How do you not make more tryptophan? You don't express these genes. How do you not express those genes? Park a negative regulator on the operator of the tryptophan operon. So when does the negative regulator need to bind? How about when there's lots of tryptophan around? Okay? Simple, straightforward, pretty obvious. Could you have come up with a simpler on-off switch or a more relevant and correct kind of on-off switch than what is going on here? There is an interesting little difference here. Notice that when the protein product of the TRPR gene is binding, it's when it's also bound to tryptophan, right? For the lac operon, remember the lac operon with P-lac-I, it's a two-trick pony, and it could only do one trick at a time. It either bound to lactose or it bound to the operator of the lac operon. The protein product of the TRPR gene is kind of a two-trick pony, but it either does both tricks at the same time or no trick, right? If it binds to tryptophan, then it binds to the operator. If it's not bound to tryptophan, then it doesn't bind to the operator. Similar, different, right? Appreciate the similarity, appreciate the difference. It boils down to the fact that the lac operon is a catabolic operon, and the TRP operon is an anabolic operon. That's why there's that subtle difference in the behavior of the repressor proteins. Check out the lay of the land. Look at this. You got a 5' UTR there. You got a start codon there. You got a stop codon. You got a start codon right after it. You got an ORF. Dare I? No, man, this is horrific. You got an ORF. Wow, I challenge you guys to write better with this little mouse thing that I've got to play with here. I may as well just get out some crayons and start scribbling. But you got an ORF after the start codon. At the end of the start code, at the end of that ORF is a stop codon. Right after that stop codon, there's another start codon. Ribosomes, as they're coming along, are going to be able to translate the TRPE protein off of this cistron, that open reading frame, and then the TRPD protein off of that one. So there's the tryptophan operon for you. A lot of stuff similar, couple very subtle small differences, but I think it gives you, between what you know about positive regulation and negative regulation from the lac operon, and now what you see with negative regulation for the tryptophan operon, you can see how you might be able to cobble things together to answer and solve different problems. What if you wanted to make the amino acid methionine, for instance? Guess what? There's a methionine operon. Guess what? It's got an operator. Guess what? There's a regulatory protein protein. It happens to be a negative regulator that picks up on the levels of methionine inside of the cell. If you have low levels of methionine, the regulatory protein comes up. Cysteine, alanine, isoleucine. Every amino acid has an operon with a set of structural genes that works just like what you see here for the tryptophan operon. Lactose isn't the only second favorite sugar for E. coli. There are others. They've got operons just like what you know now for the lactose operon. It's a recurring theme. You guys got it figured out. This is how E. coli responds to stimuli at a molecular level. Pretty cool stuff. All right, so let's move on and start talking about eukaryotes and eukaryotic regulation of gene expression. So you're a eukaryote, feeling kind of smug, I suppose, right? We're pretty remarkable organisms, awfully complicated. We've talked about this time and again in this class, eukaryotes. When you think eukaryote, think complicated. And in the scheme of eukaryotes, you guys are at the high end of eukaryotic complexity. You know, at one end are single-celled eukaryotes like yeast, right? You guys, as humans, you're not just complicated, you're like uber or super complicated, right? You're maxing out on the complexity scale. And there's cool stuff that you can do because you're so complicated, right? You can drive a car, you can make rockets that go to the moon, you can eat all kinds of different foods, you can sterilize and squash and destroy prokaryotes left and right. I mean, you guys are pretty swell. So there's good things that come from your complexity, but there's a price you pay for that complexity. You're not as nimble. You're not as responsive to the environment as something that's simpler can be. And to be able to cater to your complexity, you need to have some pretty complicated things in place. The complexity, in some sense, breeds complexity. You can't solve complicated problems with simple solutions. You need complicated solutions for your complicated problems. Regulation of gene expression is probably the single most complicated problem that eukaryotes have to solve, right? It's a bear of a problem. Let me just put it to you this way. I'll give you an analogy to help you wrap your head around how crazy, absurdly difficult gene expression is for eukaryotes. Check it out. You've got one genome. 3.2 billion nucleotide long genome. Pretty much every cell in your body has that same genome. Think of it as a blueprint. Every cell has the same set of instructions. And yet, you have trillions of cells. Pretty much every one of your trillions of cells needs to tap into that genetic information, to tap into that blueprint differently. Pretty much every one of the trillion cells needs to look at the same instructions and end up interpreting them in a different way. Some of the differences are crazy different, like the way that a brain cell reads those blueprints is ridiculously different from the way that a liver cell or a heart cell reads that blueprint. But even within the brain, there are tens of thousands of different kinds of brain cells. And even for the brain cells that are more or less the same, brain cells in the front of the brain that are more or less the same need to act differently than brain cells in the back of the brain that are more or less the same. Pretty much every cell has the same blueprints. It is awash in the same set of signals, and yet needs to read those blueprints differently. It's akin to this. You guys know that at Wright State we've been building some buildings. I think the newest building is the new classroom building. I'm sure you guys have had a chance to spend some time in there. It's what, about 3 years old now? I actually had a pretty significant role in the placement in the design of that new classroom building. I was the faculty president at the time that all that stuff was getting sorted out. And there were times when I had meetings with the architects that were involved. I kicked around the idea of saying, here, let's do this. How about this? We've got budget issues here at Wright State. We had budget issues back then. Wouldn't this be a cool thing to do? We could save some money because, I like to swim. I go to the pool fairly regularly, and it's a decent pool. But it's like a pretty old, beat-up pool. Pretty much, have you guys seen UD's pool, University of Dayton's pool? Oh my gosh. I mean, it's embarrassing when you look at our pool compared to UD's pool. Have you been to Miami universities and seen their natatorium? Whoa. I mean, you guys, whether you swim or not, you got to go check out Miami's pool. It is just jaw-dropping amazing. Question about pools?
Well, not really.
Oh. Oh, it's just massive. The ceilings are tall. There's a 10-meter platform you can dive off of. Not a 3-meter board, but a 10-meter platform. Wow, it's a beautiful pool. So I'm talking with these architects about the design of the new classroom building. And I'm kicking around this idea. Do you suppose I could ask them to do this? Could I ask them to say, what we want is a new classroom building. We want this number of classrooms with these number of seats, certain number of bathrooms and office spaces. But what I'd really like is when you give us those blueprints, how much extra would it cost if when you turn those blueprints at a 90-degree angle, now it gives you the instructions for our new Wright State natatorium. Right? How about that? Oh, and then I also want, when you use rose-colored glasses, to be able to get our indoor dogging track, so I can see how to make that out. So I just want one set of blueprints that I can make 3 radically different buildings out of. And while we're at it, I want a faculty club, I want better residence halls, I want a presidential mansion, I want all of these things with one set of plans. That's what your cells are doing with your genome, right? You get one set of instructions and you're making trillions of different kinds of cells. How do you do that? On the surface, at a first pass, you should think, that's absurd. That can't be done. What would the architect say when you ask them, give me these plans, right? They'd say, that's just insane. Nobody's going to do that. They'd think they were being really clever if they could give you one plan that could be used to make 2 different kinds of buildings. I don't want 2 kinds of buildings. I want trillions of different buildings. And there's no architect, right? This is just nature, random die roll sorts of things. This can't be done. And yet you guys are proof that it got sorted out and it's been done because you're working. It works. You got a 3.2 billion nucleotide long genome that trillions of different cells using the same sort of background inputs interpret differently. Congratulations, you're a fine piece of work. How can you solve this problem? That's the problem that eukaryotes have. Well, it's a complicated problem. And the way you solve that problem was with a very complicated solution. And despite our complexity, we have been able to sort of distill down the solution into 5 parts, right? The way that eukaryotes solve that problem is by tapping in to 5 different kinds of strategies. And each of the arrowheads in this slide, there's an arrowhead, there's an arrowhead, right? Here's one. Here's another one. Wow, this is clumsy. Here's another one. There are 5 different arrowheads there. Each of those arrowheads, in some sense, lets you focus your attention on different facets of the solution to this problem of regulating gene expression inside of eukaryotes. And to maybe put more of a point to it, the problem is tissue-specific regulation of gene expression. How do you get different tissues with the same input to give you different solutions? How do you regulate genes in an appropriate tissue-specific fashion? In prokaryotes, the game is all about being energy efficient. Right? You guys with me? Have I convinced you of that? It's all about energy efficiency. In eukaryotes, energy efficiency is swell, right? Nobody's going to scoff at energy efficiency as a eukaryote. But a much more pressing, a much more demanding problem is tissue-specific regulation of gene expression. And so for eukaryotes, when you're looking at how these things play out, I hope you'll think, whoa, that is ridiculously wasteful of energy. You're burning ATPs like crazy here. But when you see that happening, let that reinforce for you this idea that energy isn't as big a deal for eukaryotes as it is for prokaryotes. A much bigger deal for us is appropriate tissue-specific regulation of gene expression. So in prokaryotes, how does gene expression get regulated? 2 or 3 ways. At the level of the initiation of transcription, at the level of the stability of the messenger RNAs, how long they hang around, at the level of do you make the protein or not, and how long does the protein stick around. That's about it, right? Fair assessment of what we've talked about so far, more or less. In eukaryotes, we add a couple layers. We start with this idea of chromatin modification. Our DNA, so in labs this week, you guys, if you stick around, get to isolate DNA, right? The DNA that you isolate is from a eukaryote, right? But it's actually not just DNA. What you're isolating really is chromatin. And chromatin, the sort of native form of DNA inside of eukaryotic cells, chromatin is actually 2 parts protein for every 1 part of DNA. 2/3 of the mass of chromatin is protein, 1/3 is DNA. Okay? The DNA inside of your nuclei is intimately associated with a bunch of protein. And that protein DNA complex together we call chromatin. And it turns out the way that you package your DNA with those proteins has a lot to say about how you express genes. Think of it this way. You guys know I have some kids. I also am an old guy, so I've got things like encyclopedias in my house. You've heard of encyclopedias, I'm sure. There are these old-fashioned, quaint concept, right, where you'd have a book that has something interesting to say about all the things that begin with the letter A, alphabetical, and then another book that has something interesting to say about all the things that begin with the letter B. Nobody uses encyclopedias anymore, but there's still some hanging around. I've got encyclopedias in my house. I've got some kids. What kind of cruel trick would it be if for my kids, I said, you are going to learn stuff, and here's the deal. You're going to read every encyclopedia that I've got, every volume that I have, but I'm going to play a trick on you. You know, Sydney is one of my daughters. You don't get to read anything from the volume S, right? You know what I'm going to do? I'm going to take the volume S out of the set of encyclopedias. I'm going to wrap it in duct tape. I'm going to put it in a box, fill it up with styrofoam puffy things, and I'm going to wrap the box up in duct tape. And then the best part is I'm going to put it in the scariest part of the basement, the same place where I put all the Christmas presents, right? Nobody will go there. What are the chances that my daughter's going to stumble upon that box and look at it and get the information out of the volume S? She'll have access to all the rest of the encyclopedias, but she'll never know anything about the world if it begins with the letter S. That's what we're doing with chromatin, is we wrap up parts of our DNA. We bundle it up in such a way that an RNA polymerase will never venture to that scary part of the basement, right? If you bundle it up, package it really tightly, hide it away in a little dark corner of the nucleus, RNA polymerases won't ever see it. You never have to worry about expressing those genes in those cells where it's been bundled up in that particular way, okay? What I've described to you, that really tight bundling, the packaging, dark corner of the nucleus kind of thing, we got a word for that, actually. It's called heterochromatin. Heterochromatin is densely packed chromatin, bundled up, hidden away from everything else. Hard for RNA polymerases to ever bump into it and make an RNA copy that would ultimately result in a protein being produced. The alternative to heterochromatin is euchromatin, meaning open chromatin. U is EU chromatin. And that's loose, open chromatin. It's like, come on, RNA polymerase, I'm right here. Come to me, right? You know, as opposed to hard, bundled up, dark part of the basement. It's flapping around in the breeze trying to get the attention of an RNA polymerase. So we've got two kinds of chromatin, euchromatin and heterochromatin. When an egg gets fertilized, the DNA in the egg and the sperm come into this party as very euchromatic, very open, flapping around in the breeze. But with each round of cell division during human development, more and more of your DNA gets relegated to heterochromatin status. And so as tissue differentiation plays out during embryonic development, more and more of your DNA gets bundled up such that when you're a functioning adult kind of organism, even as an infant, the DNA in your brain cells is very different in terms of its packaging than the DNA in your liver cells. In your brain cells, there's only about 10 genes. Okay, there are a couple hundred genes. There's some genes that need to be expressed in all tissues at all times. Housekeeping genes, genes involved with glucose metabolism, right? Stuff like that. Genes that make ribosomes, genes that make RNA polymerases. Every cell needs those. Those will be euchromatic. But then there are some tissue-specific proteins. There's like 10 proteins that brain cells make that no other cells make. Those are euchromatin in brain tissues. But in liver tissues, that's part of the heterochromatin. And can I say vice versa? There's like 10 proteins that liver cells need to make that no other cells should be making. Those are euchromatin in liver and heterochromatin everywhere else. Okay? So how do we solve this bear of a problem of tissue-specific regulation of gene expression? Part of it is with the way that we package and bundle up our DNA. What's going to happen in the next 10 slides or so is I'll show you some pictures that help illustrate chromatin modification. I'll show you some pictures that show you what kind of crazy stuff is going on with the regulation of the initiation of transcription. I'll remind you about things I've already told you about with respect to the processing of RNA. I'll remind you of some things that are going on with respect to how long the RNA sticks around. I'll remind you that there's opportunities to translate RNAs and not to translate RNAs. And I'll show you how it is that we break up proteins once we've made it, so that once you've made it, it doesn't stick around. But that's what's going to happen. Let me show you some pictures now about what's going on with chromatin modification. Oh, the next two slides. This slide and the next one is just a zoomed-in version, more or less, on the last one. Here is a artist's rendering of what's going on with chromatin. Most of that protein that's associated with chromatin is a kind of protein called histones. Histones are very evolutionarily conserved. All eukaryotes have the same basic amino acid sequences that give rise to their histone proteins. Prokaryotes do not have histones. Prokaryotes do not package their DNA. This is a eukaryotic trick. And again, it's probably because we got this problem. But you can see they're showing you these purple things here. That's supposed to be, boy, it's amazing how hard it is for me to draw a circle. I'm going to keep doing this till I get an actual circle. So, but these are, oh, I'm going to give up. But these are histones, right? And you can see there's a set of 4 histones there. What they're not showing you is there's another histone behind this histone and behind this histone. They come together in groups of 8 octamers. You can see there are 8 tails poking out there. But around the histones, DNA gets wrapped. For every 8 histones, you wrap the DNA around the outside of that hockey puck 2 times. All right? When you're bundling up the DNA, by associating it with these proteins, those proteins, those histones, can get modified. Specifically, they can get acetylated. They could also be phosphorylated. let's focus on the phosphorylation. That'll make it easier for you to imagine what's going on there based on things that we've talked about. If you put a phosphate group on the surface of a protein, that's going to change the way that protein behaves, right? It's got a negative patch on it. And if you put a bunch of negative patches on it and its neighbors, they're going to repel each other, right? And so that's going to cause those modifications to the histone proteins are a really important step in the conversion of heterochromatin to euchromatin. All right? The more you modify the proteins, the more the chromatin loses its grip on the DNA, and the more open that chromatin is, and the more accessible it is to RNA polymerases. Okay? So you can chemically modify the histones and facilitate then a conversion from euchromatin, or from heterochromatin, to euchromatin. What's the chance that an RNA polymerase is going to be able to sniff out and find a promoter on that as opposed to that? All right? It's the encyclopedia and the scariest part of the basement thing all over again. All right? So the way we modify our histones and the way we modify our chromatin has something to say about whether or not genes are going to be expressed. Seems like a pretty good system. Then let's talk about the lay of the land for genes and the lay of the land for initiation of transcription. So just like in prokaryotes, transcription gets done by RNA polymerases, central dogma all over again. Translation is done by ribosomes, right? Information flows from DNA to RNA to protein. Here's what a gene within a eukaryote kind of looks like, sort of a generic, run-of-the-mill kind of gene. Just like in prokaryotes, there's a promoter. Just like in prokaryotes, you know what goes on at the promoter? That's where an RNA polymerase is going to hunker down and eventually, at some point, initiate transcription. Just like in prokaryotes, there is a transcriptional start site. Just like in prokaryotes, that first nucleotide to be transcribed is the +1 nucleotide. It happens to be an A. Okay? So you start transcription because an RNA polymerase has been hanging out at a promoter. When you begin transcription, you transcribe through these exons and introns and exons and introns and exons, right? We've talked about this already. There's going to be a 5' untranslated region, just like in prokaryotes, transcribed through an exon. At some point, there will be processing of that primary transcript to get rid of the introns, excuse me, and give you the final messenger RNA that ribosomes will see out in the cytoplasm, and ribosomes will be able to translate. Okay? So there's a modification that gets made to the 5' end. We've talked about that, the 5' cap. There's this internal modification, the removal of the introns by spliceosomes. And then there's also the addition of a tail of 200 or so adenines, the poly-A tail. This is kind of new and different relative to what we had talked about for prokaryotes. That stuff happening over there. There's a couple other things that are new and different. The introns doesn't happen in prokaryotes. You guys remember why, right? Transcription and translation happen at the same time in the same place for prokaryotes. In eukaryotes, transcription happens in the nucleus. Modifications like splicing happen in the nucleus. Translation happens only in the cytoplasm. Okay? So there are already some differences. But the stuff going on the upper left part of this screen here, that's different than what was happening within prokaryotes. Remember those pictures of the lac operon and the trip operon? Do you recall, did you notice how much real estate at the DNA level was set aside for regulation of the lactose operon? Stop and think about that for just a few seconds. You know, how many nucleotides are necessary for the machinery to be able to appropriately express the lactose operon, to key in on lactose levels, to key in on glucose levels. Right? There's the 30-some nucleotides that are part of the promoter, right? Minus 35, minus 10, plus 1, 30 or so nucleotides there. Only a baker's dozen get looked at, but nonetheless, that's how much real estate's being taken up. Downstream of that, for the lac operon was an operator. Triplet operon as well, there's an operator. The operator, I've said 20 nucleotides or so. The whole package, beginning to end of those regulatory sequences, 60 nucleotides tops for a typical prokaryotic operon. Okay? Not that much to ask, not that much real estate. When you got a genome that's 4 million nucleotides long, setting aside 60 for regulatory purposes, yeah, that doesn't seem too wasteful. Not a bad way to go. With eukaryotes, of course, you're going to need the same sort of real estate for the promoter, right? You're still looking at about a dozen or so nucleotides. There's the plus 1 nucleotide. Everything's spread out a little bit more. Instead of at minus 35, there's something at minus 80, right? So it gets spread out a little bit more, but not that big a deal, right? So there's minus 80, minus 40, plus 1, all that stuff, 80 or so nucleotides. But the new thing here is all of this upstream stuff, this enhancer, the distal control elements, the proximal control elements. You know what this means? You guys know what this means. That means that if they drew this to scale, you wouldn't see anything. There's a big gap here. These upstream control sequences, get ready, they can be tens of thousands of nucleotides upstream. Okay? Let me tell you a horror story that I got to witness. So as a graduate student, I'm working in a lab with other people. One of the other people in the lab was there, I think she got there a year before I started. And so pretty much every day when I'd walk into lab, this woman, Magda, short for Magdalena, but Magda would say to me, it's like, oh, so Dan, when are you going to graduate? When are you going to be done with your PhD work? And whatever I came up with, she would always throw back at me, well, I'll be a year faster than that. I've been working at this for a year longer than you have. Yeah, fine, Magda, I get it. I got the joke. You know, every day we'd go through the same thing. Magda's thesis project, what she had to figure out is how it is that rabbits, interesting story in and of itself, but how it is that rabbits appropriately express the beta globin gene, one of the 2 proteins you need to make hemoglobin, in red blood cells, and only red blood cells, right? And the thinking back then, in the old days, was that whatever rabbit blood cells we're looking at was probably just upstream, because all we knew is how things worked with the lac operon. And what she would do is she would take chunks of nucleotide sequences from upstream of the transcriptional start site for the rabbit beta globin gene, and she would try to put those chunks of nucleotide sequence in front of other genes and then get those back inside of rabbits and look to see if she could trick red blood cells in rabbits to expressing genes they shouldn't be expressing. So she'd take thousands, ultimately tens of thousands of nucleotides. And the funny part of the story, from my perspective, not from Magda's, is that I got a PhD. And she was still working away at trying to figure this out for about 5 years after I left, before she gave up. All right? And so 10-some years of somebody's life was spent trying to figure out, well, how do rabbit cells know this is something that should be expressed only in red blood cells? It turns out we now know the answer. The answer is that the sequences that rabbit red blood cells are looking for that tell it this is a gene that should be expressed in red blood cells and not anywhere else, they're not just a couple thousand or 10,000 nucleotides away. For rabbits, they are 65,000 nucleotides upstream. There's a stretch of little tiny patches enhancer, distal control elements, 65,000 base pairs upstream of the transcriptional start site that determine if the rabbit beta globin gene will be expressed in red blood cells as opposed to anything else. 65,000 nucleotides. You guys don't seem suitably impressed by that, right? Put this in the context of E. coli again. You know, the regulatory sequences at E. coli are a couple dozen nucleotides, right? Here we're talking about on the order of 100,000 nucleotides. 100,000 nucleotides, for goodness sakes, E. coli's genome is only 3.2 million nucleotides long, right? 100,000, that's 1/32nd of the entire length of E. coli's genome is set aside just for the purpose of regulating one gene in the rabbit genome. 1/32nd of the entirety of E. coli's genome is what you use and you're just playing around with to be able to get regulation of the beta globin gene in rabbit red blood cells. And it turns out it works out the same way for human red blood cells. Rabbits and humans are playing the same game with that particular gene. Lots and lots of upstream sequences. The best estimates are that for every mammalian gene, there are 7 things that need to be in just the right conformation in order to get a gene expressed. 7 thumbs up have to be delivered before you can persuade an RNA polymerase to start transcription. And those 7 things can spread out over the course of tens of thousands, maybe 100,000 nucleotides. In prokaryotes, you've seen it with the lac operon. There are 2 things that need to give a thumbs up. You need both to have low glucose levels and high lactose levels. For eukaryotes, you got not just those 2 kinds of things, you got a whole bunch of other stuff, like 5 other things, and they can get spread out over an enormous amount of real estate at the level of DNA. And I've already talked with you about how intrinsically wasteful the transcription of introns can be. You know, literally millions of nucleotides of introns for a single gene, burning ATPs left and right. Apparently, it's a price we're willing to pay so long as we can be sure to never express a liver protein in your brain. Because when that happens, apparently bad things happen, like tumors and death, right? So to solve this bear of a problem, we're willing to burn ATPs at the level of how much DNA we need to lug around and replicate. We're willing to burn ATPs in terms of how much DNA we transcribe, how many introns we end up just chucking for no good reason. We have very complicated promoters. Okay? Here's another way of looking at the splicing business. I mentioned this to you guys already, but I don't think I showed you this particular picture. I told you about poor Dick Cheney and his heart attack and cardiac myosin. You might recall that story. You've got about 25,000 genes within the human genome, 35,000, depending on who you talk with. You've got a decent number of genes, but you can make 250,000 proteins, like 10 times as many different proteins as you have genes that could code for them. The reason that you can do that is because of alternative splicing. Here's a pretty picture that shows you for a particular gene, troponin T. It's a gene with 5 exons, only 4 of which end up in the messenger RNA. In some tissues, troponin T is made with the coding information from a 3rd exon. And you never see the 4th exon as part of those messenger RNAs in those tissues, but in other tissues, the 3rd exon gets spliced out as part of an intron, and you end up only with the 4th exon in the messenger RNA, and you have a single gene, but you make 2 fairly substantially different kinds of proteins from it. So alternative splicing is in play here. One other thing for tissue-specific regulation of gene expression. I suppose you could, in some sense, have tissue-specific genes. So let's talk a little bit about the proteins that bind to DNA. There's so many things I'd like to share with you guys in a class like this, and there's not enough time for me to tell you all the stories. Some of you may be wondering how proteins sniff out information at the level of DNA. This picture answers that question for you, if you have that question. If you think about what a DNA molecule looks like, that classic double helix sort of thing, the most interesting bits of information are right where the hydrogen bonding between one nucleotide and another nucleotide takes place. That's where you get the most distinctive features. That is, as I've already said in this class, the dance cards for those features are full. They're already involved in hydrogen bonds, and so there's nothing really that can be seen there. The next best thing that you can do is lay a protein across what's called the major groove of that double helix, lay it in there with an alpha helix, and let the amino acids on the surface of the alpha helix sniff out features on the edges of the nucleotides. And so that's exactly what's happening here. You guys probably recognize that as an alpha helix. It turns out that most DNA binding proteins, both in prokaryotes and eukaryotes, work as dimers. So two copies of the same protein. Here they're showing you one dimer in purple, the other dimer in a salmon, sort of pinkish color, but they each have an alpha helix, and they're laying down their alpha helices right next to each other, one on one side of the major groove and then the other on the other side of the major groove. And then the other parts of the proteins come together at the top to either attract in another protein or something like that. So that's where the action's taking place. Look how many nucleotides that alpha helix could sniff out. It's getting some information from 1, 2, 3, 4, maybe 5, maybe 6. It's unrealistic to expect a protein to be able to, a single protein to be able to interact specifically with more than 5 or 6 nucleotides. Does that number ring a bell? That's how many nucleotides are in the Tata box and the Pribnow box. You know why? It's because that's as big of an alpha, that's as much as you can expect an alpha helix to do. All right, so same thing over and over again. And it turns out that a lot of the protein binding sites are what are called palindromes. They read the same forward as backwards. This alpha helix is the same as this alpha helix. What they're looking for here is a set of nucleotides that reads in one way down this axis and then reads in the same way down that axis. So anyway, that's what a DNA binding protein looks like. DNA binding proteins play a big role in terms of initiating transcription within eukaryotes. There are these activator proteins that have to all be in place before you could have anything taking place at the promoter far downstream. 60,000 nucleotides away. Turns out to be able to get that to work, you have to have some other proteins that come in that bend the DNA molecules so that you get those promoter sequences in proximity to the activator proteins. And it's only when all of this stuff is in place, protein here, protein there, stuff going on over here, the bending protein, only when all of those things are in place do you have a chance to have an RNA polymerase initiate transcription and make a copy of a gene. So what we got here is a pair of genes, right? We got the albumin gene, a protein that's part of our blood made by our livers. And you've got the crystalline gene, a protein that makes the lens crystal in your eyes. They're showing you these two genes side by side, but what they want to draw your attention to is the difference in the enhancer sequences, the control elements between these two genes. Again, tens of thousands of nucleotides can separate where the enhancers are relative to the promoters. But in liver cells, apparently liver cells are awash with these Mickey Mouse hat molecules. You know, so it's easier to see there, all right? Those stick to the red, so they have a DNA binding domain there that lets them stick to that, right? When they stick, then in a liver cell, you can assemble all the rest of these things that lets you initiate transcription. But in a lens cell, you don't have a red binding site for the crystalline protein. You've got a pink binding site. And since there's no pink things floating around inside of the liver cells, you don't bother to initiate transcription of this gene. And the converse takes place then inside of a lens, inside of a lens cell as opposed to a liver cell. So part of the way that we turn on genes in some tissues as opposed to other tissues is packaging, but part of it also is what sort of transcription factors, what sort of enhancers are provided inside of one tissue type as opposed to another tissue type. All right. So you got that. Let's get to the home stretch here. In earlier lectures, I've talked with you about the idea that messenger RNAs don't stick around forever. Once you've made an RNA copy of a gene, it needs to get broken down. I suggested to you there's a popular school of thought that says that the longer the poly A tail is, the longer the messenger RNA sticks around, the more proteins you can make from it. Enough said about that. Let me also now just give you some details about how long proteins stick around. Okay? Because once you've made a protein, you short circuit the whole gene expression process if you keep the protein around forever. In eukaryotes, what happens to a protein after it's been used for a while is it gets ubiquinated. What that means is a molecule called ubiquitin gets tacked onto it. The ubiquitin causes the protein to attract the attention of a proteasome. And when a protein enters into a proteasome, it's sort of like abandon all hope, ye proteins who enter here. It's a meat grinder. And what happens is the ubiquitin gets released unharmed, but the protein gets broken up into little tiny peptides that can then be broken down to their component amino acids and used to make new proteins with. Different proteins get ubiquinated at different rates in different tissues. How about that? It's worth contemplating a little bit. One tissue type may ubiquinate a particular protein really fast and give a pass to another protein. What's going to happen? Even though you've expressed the protein, you'll have low levels of that protein that got ubiquinated relative to the one that didn't get ubiquinated. In a different tissue, a different cell type may play the game differently and change those ubiquitinization rates. And the circumstances under which each cell finds itself may also have an influence about how long the protein sticks around. So we can play games with how we package our DNA. We can play games with what genes we transcribe. We can play games with what genes we translate once we've transcribed them. We can play games with what kind of messenger RNAs we make. We can play games with how long the protein sticks around and how long an RNA sticks around. We take full advantage of every one of those things to pull off that trick that we have to accomplish of tissue-specific regulation of gene expression. And we're willing to throw away millions of ATPs worth of work at each step of the process, just so long as we never have the problem of making a liver protein in a brain cell, and vice versa. All right, well, I think that's plenty enough for today. These last two slides are simply giving you a little bit of a closer view of the things that I've just been talking with you about. Let's call it a week and come back on Tuesday of next week when I'll talk with you about how it is molecular biologists figure stuff out, what tools we have in our toolbox. Have a good weekend. See you guys Tuesday.